The other way is breaking symmetry is by having different environments. So the idea is that, again, you've got your mother cell, you've got two identical daughter cells, but this daughter cell is in a slightly different environment than the other one. It's being impacted by whatever factors are here, and that causes that cell to be slightly different. So again, breaking symmetry is all about going from one cell to two different cell types. How you do that, whether or not it's because there is some sort of intrinsic difference between the two daughter cells, or whether it is that one daughter cell is in a different environment than the other one, you still end up with the same thing happening. Any questions about that? Yeah?
So I can only assume that there are proteins that attack the second daughter cell and that move it to another environment.
So remember that these cells are motile, they can crawl.
Okay.
So yeah.
Because I only assume that if they first divide, I mean, something has to happen in order for them to.
Right, so either there's a higher concentration of some chemical down here that's not here, so that's our response, or this cell crawls to a different place. But again, this idea that you can get this break in symmetry by having a differential impact of the environment on one cell and the other. So then once you get that initial asymmetry developed, what you want to do is you want to amplify that. Because the thing is that when you're going through development, you don't want to have two cells that are slightly different. You want two cells that are vastly different. And so the way that you can do that is a process called lateral inhibition. So the idea is that if you've got two cells that are relatively close to each other in type, and they are each producing this protein, in this case protein X, each one of these cells is inhibiting the production of X on the other cell. So that's where the inhibition is coming in. Lateral being side by side, inhibition. It ends up that, for one reason or another, there's a slight difference between one cell and the other. So this cell makes more X. It's able to have a stronger inhibitory effect on this cell. Therefore, this cell makes less X and inhibits this cell less. And so you get this positive feedback loop where this cell is able to make more and more X. This cell is making less and less X, to the point where you've got this cell that's making tons of X, and therefore is inhibiting production of X in this cell. This cell can't make X, and therefore cannot inhibit X production of this cell. So you end up getting these two different cell types. So again, you're getting this all or none effect. Again, all due to this feedback loop that gets set up by this lateral inhibition. Any questions about that? So again, it's this idea that you start off with a very, very slight difference created by breaking asymmetry. That asymmetry gets magnified through lateral inhibition. Any questions about that? So then another way that you can magnify asymmetry is through the use of morphogen gradients. So again, the idea being that you've got cells of a certain type, and they're producing some signal, some morphogen. And that morphogen diffuses away from these signaling cells. And since these cells are nearby a high concentration of morphogen, they are going to undergo a certain developmental pathway. Cells that are further away from the source of morphogen don't get enough of it, and so they don't induce that pathway. So again, you're able to cause and magnify any sort of asymmetry between this group of cells here and this group of cells here, simply by using the fact that these cells are bathed in a specific morphogen, a specific compound, and these cells aren't. Any questions about that? All right. So then there are a number of ways you can produce a morphogen gradient. The simplest is that you have one set of cells that are producing morphogen, and it diffuses away. So you end up with high concentration of morphogen here, low concentration here. These cells that are in this area of the gradient are going to be exposed to it, are going to undergo one differentiation pathway. These cells are not, they're not going to. The other way that you can do it is you can have the morphogen be expressed equally throughout the field, but you have the presence of cells that are producing an inhibitor of morphogen. And so cells that are this part have a high level of inhibitor, and so the resulting amount of morphogen present is low. These cells that are further away from the inhibitor have a low level of inhibitor, therefore their levels of the morphogen are high. So you get this morphogen gradient being produced even though the morphogen itself is evenly distributed. So again, there's just these number of ways you can actually end up getting a morphogen gradient. It doesn't matter how it gets set up, it's going to have the same effect. Cells here are going to develop one way, cells here are going to develop another. Any questions about that? All right. And so then, you can see then that you could be able to get these different types of cells differentiating by having these different waves of induction. So again, this idea that you might start off all with cells of one cell type, and then through the use of breaking asymmetry and morphogen gradients, you end up with two different cell types, cell type B, cell type A. Cell type B might be sending a signal that causes the production of cell type C. So now C, because it's a different cell type, can start sending out its own signals to cause A and B to differentiate to different cell types D and E. So again, you get this branching that I talked about, where you get this sequential differentiation, again, based upon the basic things that we saw before, of setting up this initial symmetry breaking, and then reinforcing that asymmetry. We end up with multiple different cell types. Any questions about that? Clear as mud? Great. Okay. Okay, so get out your clickers. All right, so what type of signaling would be best for morphogen? The majority of the people said C, paracrine, and that is correct. So why is that? Why is it not cell contact? Well, if we go back and take a look here at how this works, again, what we see is that the factor, whatever it is the inductive signal is, has to diffuse away from the point of its origin. So it cannot be cell-cell contact, because cell-cell contact requires the two cells physically interact with each other. So if it were cell-cell contact, these cells over here would never get the signal. So it can't be cell-cell contact. So why is it not autocrine? Well, it's not autocrine because remember, autocrine, while it does deal with the diffusible factor, autocrine means that it's impacting itself or cells of the same cell type. And so you can't have a morphogen affecting itself. The idea is that the morphogen is causing a differentiation of a different cell type aside from itself. So there must be a difference between those cells that are inducing the signal and those cells receiving the signal. Otherwise, instead of getting two different cell types, it'll all be the same cell type. So it cannot be autocrine, even though it's a diffusible factor. Therefore, it's paracrine. So paracrine is a diffusible factor that's impacting a different cell type. And so that's exactly what we're looking at. We're looking at the fact that the cells that are producing the morphogen gradient are different than the cells that are being impacted by it. Synaptics only involved in neuronal cells, and endocrine is over long distances through the bloodstream, so it can't be those two. Any questions about that? All right, so then for the next question, how would you narrow the effective impact of a morphogen? So if you wanted to make sure the morphogen only affected very thin band of cells, how would you go about doing that? By decreasing the diffusion rate and increasing receptor affinity, by increasing the diffusion rate and increasing receptor affinity, by decreasing diffusion rate and decreasing receptor affinity, by increasing diffusion rate and decreasing receptor affinity, or none of the above. I'm talking about the receptor being able to bind to the morphogen. So looking at the Katie. Right? So majority said A with a close second being C. The correct answer is C. So you want to decrease the diffusion rate and decrease the receptor affinity. So why do you want to do that? How does that work? So again, the idea is you want to decrease the diffusion rate, meaning that if this is your cell that's making your morphogen, you don't want the morphogen to diffuse far away. You want the morphogen to only diffuse a little bit, so you want to decrease the diffusion rate. And you want to decrease receptor affinity. The idea being that, again, if this is your cell making your morphogen, let's say you've got a set diffusion rate. If you have a high affinity receptor, that means that very low concentrations of morphogen will be detected. Meaning that cells that are even very far away will be able to sense the morphogen. However, if you've got low affinity, you're going to need high concentration of the morphogen to be detected. So these cells back here aren't going to detect the morphogen because it's not at a high enough concentration. Only those cells that are close by the source will detect it. And so again, you're narrowing the range in which the morphogen can have its impact. Any questions about that? All right, so that's it. What we're going to do on Thursday is give some real-life examples of how this works in different organisms, and then finish up our lecture on adhesion. Last thing I want to say is that I have also posted last year's exam, or previous year's exam, just to give you an idea of the types of questions that I asked. There's material on that exam that you guys, we didn't cover, so you're not going to be responsible for. There's also information that we have covered that's not on that exam, so this is not there because you don't have to know it. Lastly, I've also posted a journal article, basically about the cytoskeleton and motility. So please go ahead and read that. We will talk more about what my expectations are from you on that